Joic, welcome, and it's a pleasure to have you here, which we tried to schedule this for a long time, and we finally sit down here. So welcome.
Thank you, Luis. Yeah, it's good to be here, and thank you for having me.
No problem at all. Were you at university this morning?
Yeah, I've been doing some work in the office today,
Okay,
a little bit in the lab.
okay, what was it about?
So we work with human brain tissue,
Mm-hmm.
and we have like little blocks that we have to process, stain, and at the end you have to mount it with a little glass slide on top, so
Mm-hmm.
then it's fixated.
Yeah.
And I had to do that on like our last experiment.
Oh, wow. So was there any concluding results already, or is it still on hold?
So we do something called digital pathology,
Yeah.
and it's like you basically do that experiment and then you send the slides to a center where they have a very fancy printer or
Mm-hmm.
scanner, and
Yeah.
then they put all those images, very large files, into a drive.
Yeah.
And then we receive the drive and we analyze them um, digitally, using a program, and then maybe we find results after.
All right. So from what you said, for now, it is still on hold. The any outcome, any positive outcome, or maybe negative outcome, will come in due time. Then.
Yeah, it's like a multi-stage process. So the first stage is the lab. That's
Yes.
been done. The second one is obtaining those digital images and analyzing them. And at the end, maybe using like clinical information to get some
Oh,
outcomes. Yeah.
okay. So that's great to know. And I asked this follow-up question because I'd like to know, from all the conversations that we were having outside of the lab. I've been into the lab before. We're talking about our research, but I would like to know a deep, more thoughtful answer of yours of why, where this passion about researching multiple sclerosis comes from, and how that passion reinforced your thoughts in pursuing a PhD.
That's a good question. I did medical biochemistry as an undergrad degree, and there were some modules that I really enjoyed. One of them was neuroscience. So neuroscience is very interesting. It encompasses a lot of different conditions, and some of them are what we call neurodegenerative conditions. And these are things like Alzheimer's, Parkinson's, but so is multiple sclerosis. And I discovered that they're conditions that currently have no cure.
Yeah.
Their mechanisms are very intrinsic, so they're very, there's a lot of research going into them to understand why people get Alzheimer's or Parkinson's or multiple sclerosis. We say MS.
Yes.
And initially I did a master's on investigating Alzheimer's a little more, and it was a very computational master's. And then I was applying to PhDs because I liked the topic of researching these conditions that have no cure.
Yeah.
I thought that was like a good thing to do, and it was an interesting thing to do to me as well. And I ended up learning about multiple sclerosis. And you've been to the lab. That's the neuroscience floor.
Yes.
And I was always on like the left side of that lab because I've been doing Alzheimer's research, computational research, and then on the other side of the lab there was my current supervisor, Owen Howell, and I met with him, spoke about the PhD, and I was very interested, gave an interview, and I didn't get it, but I was on the on
You
the
didn't get
reserve
it
list, and the person who actually the
at the
person who got it
yeah.
for some reason dropped, and then they contacted me and they said, do you want to do it? And I said,
Oh,
now,
so it
now
was,
here we are. Yeah.
wow. So it was like a great omen to you, and or maybe sheer luck I guess most of us is like it's like sheer luck, and but that's a
I
good.
think a
That
lot
was
of
great.
stuff in life is
Yeah,
luck.
yeah, exactly,
And
exactly.
my PhD,
Mm-hmm.
just to say, my PhD is also, I do like teaching. So I got into this because of that as well. So my passion is also, I enjoy that aspect
Yeah.
of the teaching, and that's why I applied to this specific PhD.
Wow. So quite some ups and downs in the process, and having the masters would have you like as a backup plan, and maybe that would reinforce that passion of yours into multiple sclerosis or MS, as you like to shorten it, into a PhD. And I do wonder, our from a humanities perspective, our type of interview is a, not say formal and informal, so it's in between, where we have a chat with our future supervisors, right? My supervisor was the one who I had from my master's, and we were talking about doing a PhD, and the way of interviewing the application when I did it was through a project proposition. So I would like to ask you, how different is an interview for a PhD, a STEM PhD? Is there any differences? Do you also STEM candidates for this process too?
I think the core process is the same, where perhaps in STEM they usually, like the principal investigators, the PIs, they apply for grants and they
Yes.
get some money, and then they say, okay, this money is for 1 or 2 PhDs, say, or like a PhD and a teaching a lab assistant or something. And then that PhD has like a job description, so then it gets advertised, you read that job description, and then you sort of see how you would fit that description, right? And then during the interview, maybe you prepare a couple of things that you would sort of I don't know, maybe there's something that you're really good at or that you've done in the past that, it's in the description, so you bring that up, kind of thing. Mine was like a PowerPoint presentation of how I would fit that role, for example.
Yes.
So I prepare for that. But I think not everyone, but if you do get to meet I got to meet Owain before, and we had a conversation about it, and he helped me see my different strengths, that sort of how they would fit into the project. So it's really good if someone is able to, I think my supervisor is very good at, no matter what are your, like, considering the different strengths that you have, how that fits into the project, right?
Yeah.
Because some supervisors might be, I want a person that will do this lab project 100%, sort of has to be like lab background, very confident, whatever. Whereas other people might be a little bit more flexible and sort of based on, the vibes maybe, or like your different strengths, would they sort of enhance the team, would they get this done? But to make it short, it is very similar, and perhaps a little bit more guided to like where you spend this money, will you spend this money?
Yeah. Which is quite interesting, and a bit it relies on being cautious of who's more apt for these types of funded projects. And as you said, it was by pure luck, as you said, but also you would probably say, of if you are comfortable in doing intensive lab research, if you are capable of hold your grounds and trying to elevate yourself into the next step of different lab challenges that will happen throughout your PhD, do they rather count more on that, but as alongside your expertise, meaning that how comfortable you are?
Yeah, so they do try to get a sense of the person,
Mm-hmm.
not just the CV, for example, or the experience. They want to know, will they bounce back from problems along the PhD, or will they fit into the team,
Mm-hmm.
and I think that's a good thing to consider, because someone might be perfect on paper, but then once you meet them, they might not sort of reflect that perfectness. They might be like, I don't know.
So,
Yeah.
did you learn from that after your first attempt in your interview?
Yeah, I mean, I felt very comfortable with my interview because I knew a couple of people. They were familiar faces.
Yeah.
But I tried to sort of bring my strengths to the project, because a lot of it involves laboratory skills.
Yeah.
And I did have a little bit of experience, but as I said, my master's was very computational. So, for example, I said a lot of, I said exactly this, but I also added that my computational background could help investigate this and that, because I know that other than, what this is the project, there's also a lot of data that, we can use. And that's actually a lot of the things that I do now, is a lot of data analysis.
Yeah. Including the gaps and how you would turn them over, how would you provide a different solution for these research gaps?
Yeah. So we have a lot of like a big data set of neuropathology data, and we also have some clinical data, and it's like marrying both up, sort of see the relationships,
Oh, so it seems like of all these adversities, I've managed to get into a new PhD. You're on your second year, I would assume. Sorry.
Yeah.
But take us back to the first stages of your PhD. Telling us what multiple sclerosis is. We've heard, most of us have heard the word, but the study itself seems more private inside of the lab, and it's not wildly amplified out there from a scientific perspective rather than a media perspective. What is multiple sclerosis, and what are the patterns that are identifiable and determine? So this is a cause of multiple sclerosis.
Yeah. So multiple sclerosis is a chronic progressive neurological condition that affects a lot of young adults. So the onset is around 35 years old, and it affects something called a type of lipid or fat in your brain called myelin. Myelin insulates myelin, and myelin sort of insulates and wraps around the axons, so of your neurons, of your nerves. And it's also an autoimmune disease, because these myelin, they're called myelin sheaths. They belong to you, right? That's your part of your cells, and for some reason the immune system, your own immune system, degrades that myelin and starts to eating it up. So then you end up with demyelination, and you end up with a lot of inflammation, and this sort of triggers a lot of different reactions, triggers cell death. So that's why it's also a neurodegenerative condition, because your neurons and your axons die, and this leads to someone having multiple, like, different symptoms, because depending on what part of the brain is affected, but usually it's like a lot of motor symptoms. So you may need like a crane to walk, or eventually a wheelchair. You might lose functionality in your arms, you might lose some vision, some hearing, and
So
the patterns. Yeah, sorry.
yeah, go on.
In terms of patterns, what I sort of so I work on neuropathology, and neuropathology is the study of the of the brain and spinal cord, and we use post-mortem tissue, so tissue of donors that they've had the disease and they've lived with it and donated their organs. And so we study the brain, and we look at demyelination, we look at lesions, so these sort of like in parts of the brain you would have a part of the brain called the white matter, which is white because it has that lipid myelin, and then the gray matter, which isn't, because it has just the cell bodies, it doesn't have as much myelin. And then the white matter, sometimes you have holes in it, and those holes are demyelination, and they're also called lesions. And these lesions can be very active. They can have a lot of immune cells in them. So those are the sort of things that we see.
I wanted to ask about, well, you said about you're exploring the spinal cord and neuropathology in the spinal cord, the different fluids of the brain, white myelin, and then the gray area. But I like to ask, where do we see the where is more, I would say, where is more prevalent? What part of the body is highly likely to suffer from multiple sclerosis? So basically, in other words, does it start? Is it more relevant? This phenomenon, is this more relevant in the arms, on the legs, in one part of the chest? Where is more highly likely to have those symptoms?
Okay, so those lesions that I described, they can happen anywhere in the central nervous system, and the central nervous system is the brain and the spinal cord. So
Yes.
anywhere, any part of the brain, any part of the spinal cord can be affected, and that leads to the different symptoms that someone might manifest. And one thing that it is true is that a lot of the times the most common symptom is loss of vision, because because the nerves that sort of connect parts of the brain to your eyes, they get demyelinated, and then you have optic neuritis, it's called. And now this links to the diagnosis. So the diagnosis is like quite complex, but one of the signs is to look out for this optic neuritis. And then if you have it and you show other symptoms, then you might go into MRI, so magnetic resonance imaging machine, and then to see if you get the diagnosis.
Okay. Say, for example, if a person starts to feel ill, and is not knowledgeable about that multiple sclerosis may be the cause of it, can the person know, the person who's suffering from multiple sclerosis, can the person know when he or she starts to get those symptoms? Is there any sort of knowing how it starts?
Well, first of all, I'd like to say I'm no doctor.
Yes, I know.
Medical advice. But I think if you feel ill, I would say go to your GP.
Yes.
And go from there, right?
Yeah.
But it's tricky. What sort of something that is very characteristic of multiple sclerosis is, as I said, the loss of vision, but the concept of relapses, right? And a relapse is like an immune attack that can last from hours to days. So say from like you suddenly cannot walk properly for like a day, right? A whole day you don't walk properly, and then suddenly for the following week you recover that ability, right? And this is like the very traditional presentation of MS. It's called relapsing remitting, where you have a relapse, so your disability increases, and then you have like that remittance, right? Where your you recover that ability, right? So that's something to look out for. But then again, I'm no doctor. Go to your GP, and then But it is quite complex. Some people get a relapse and then they never ever get it again, and that's called clinical isolated syndrome, because that's you've had an episode, but you've never developed MS, right? Some people, a percentage of people with CIS, they do get MS, but why is that? People don't really know.
For which there's still a cure to be developed, as you said at the beginning of the talk.
Yeah.
And following that question, and how is it somewhat different from ALS or amyotrophic lateral sclerosis? Because
so ALS, they are different conditions. ALS is a motor neuron disease. So it affects motor neurons. It's a specific type of neurons that sort of, you do get these motor symptoms. So you do also lose ability to move, to walk, and it is different. I'm not an expert in ALS, but there are different markers. Even at the neuropathology level, there's different patterns, and sort of ALS is very aggressive. So usually people live maybe 5 to 10 years after diagnosis. For MS now, even though there are no cures, there are something that we say disease modifying therapies, or DMTs. And these are specific drugs that slow down the pathology. So with MS now, people can live up to their 70s, 80s, almost like a normal life expectancy.
So go on.
Yeah. So the no cure part is that some people still get progression. Progression is when you just get worse and worse, and you don't have that sort of recovery anymore. And for that, there's no drugs available, Some maybe show like a little slowdown to that point, but once you develop this progression, it's difficult. It's there's no treatment.
So it's more of a chemical therapy in that regard, because as you said, we're still far behind of finding a cure for these symptoms and right. So as you said that there will be no cure, and it is prescribed medicine. It is a way of slowing down the rapid progression of MS in this case, in order for the patient having a prolonged life and having more of a healthy lifestyle. But as you said, it's a bit complex, and that doesn't guarantee that there is a total reverse from it, or is there a reverse process aside from the prescribed medicine?
I don't know. It's like quite a dynamic, like neurodegenerative conditions are dynamic, right? Progressive conditions.
Yeah.
Because you have to think that there is the pathology. So there is like what I was describing before, demyelination, where myelin gets degraded.
Mm-hmm.
There is also remyelination, and that means some cells called oligodendrocytes they come to the neurons and they try to remyelinate, to produce myelin around that like naked axon. And then you have inflammation, right? So pro-inflammatory activity in certain like areas of your brain, but then you have clearance and, you know, anti-inflammatory signals and cascades that try to clear that inflammation out. So it's very down to someone's maybe genetic background, resilience factors. It is dynamic, you know.
Does family inheritance count as well?
I think there is a degree of inheritance, but it's not confirmed. That's another field of MS that's very not unexplored, because a lot of people are looking at it and have been looking at it for a long time. But onset of like, why do you get MS is still a little bit, they're not too sure, you know.
So it's a bit untouched waters, and it's still it becomes complex.
Therapy. The leading hypothesis is it's to do with a virus called Epstein-Barr virus.
Epstein
that have had it.
Epstein-Barr virus. Epstein-Barr virus.
Epstein-Barr virus. Yeah, EBV.
Oh, okay. Not that Epstein we know.
Not him. There's a very like reference study of I think the American military or something, because they get blood tested every 6 weeks or something, or every month. And for like these 20 years longitudinal study, they saw that those, like a portion of those soldiers who were positive for EBV then developed multiple sclerosis. So that's like the biggest sort of proof, you know. But then newer studies show that not everyone that has had EBV, so not everyone with MS has had this EBV infection. So it's still sort of, you know, a little bit in the air.
Yeah, it's still, well, as you said, it's still an ongoing and complicated study, because every human being has its own pathology. Every brain is a different world, as my father used to say, and it is complex, and it requires careful study of each one's brain in order to identify the causes of the pathological causes that led to multiple sclerosis. But then getting a solution from it, as you said, prescribed medicine to everyone is more or less an assumption that everyone has the same pathological condition, and in order to delay the process, everyone receives the same chemical components in order to slow down the pathological progression. I'm probably wrong on saying, so throwing things in the air, but would it be more or less like this when debating these sort of complex issues?
Yeah, no, that's definitely like, yeah. So what you're saying a little bit is like personalized medicine, right? Where
yes,
everyone gets assessed and then treats it for what they're suffering from. And in MS, there's a little bit of work like that, right?
Mm-hmm.
And some of the work I do could lead to that, because we look at pathological patterns of like a big cohort of people, around 300 cases,
Mm-hmm.
and then some of them are very severe because they died very young.
Oh, wow.
And then the pathology shows that there's, say, a lot of inflammation, right? A lot of like damaging lesions. So for those people, then we want to understand the nature of those lesions to maybe say, oh, these, like, there should be drugs targeting this, you know? And that maybe is helpful for that cohort of, or sub-cohort of the MS population that will develop that type of MS that is potentially very detrimental, right? So it's sort of like you work with, you have to sort of target may be the worst subgroup, you know.
When you said died very young, and backtracking to what you said, that it's usually identifiable from 35 years old onwards, were there any other cases of people who suffered from multiple sclerosis before the age of 30 or the age of 35? Were there any sort of cases you've encountered with?
Yeah, of course. Yeah. So the average symptom onset, so that's when someone had their first symptoms, you know,
Yes.
is around that age.
Mm-hmm.
But some people might have it like, I don't know, 30, 25, you know. And there's even, you know, in the cohort that we're studying, there's like a 9-year-old. But then as a concept, pediatric MS also exists. And that means, I think before the age of 18, maybe. So yeah, some people get symptom onset before that. Very young, unfortunately.
Yeah. So take us then to your lab research. You told me that you had one today and it's still going on. But did you have also any other results? So you're doing the microscopic tests, perturbations of the brain, spinal cord tissues, anything related to the brain pathology, and then the results weren't the ones you wanted. So how did that happen, and what did you learn from it, and how did you turn around to get what you want, what the results you desired?
With my results, so I'll talk about like, so a lot of the work I do is to do with the data, right?
Yeah.
And with the data, I'm trying to model this disease according to the patterns and the things that we can measure in the brain. And a lot of the times, maybe I get like some assumptions wrong, so I have to revisit revisit it with my supervisor, ask advice, and then remodel what I've previously done. Or a lot of the times, if I'm doing statistical models, I'm developing expertise, but I'm not an expert, right? I'm still sort of learning.
Yeah.
So I have to ask for help to other people that actually know their statistics and make sure that I'm doing the right things, you know, the right processes. There's a lot of things that you have to check along the way to sort of say, okay, this has been done properly, you know. So fortunately, I have a couple of people that I can show it. Like, one is on the same floor. He was also involved in my master's. Dr. Carl Ostimutir, shout out to him. He's awesome at what he does. He's a wonderful person, and he's always like open to just listen to what I have to say.
Oh, that's nice.
Discuss. And then have a collaborator.
Could be an eventual talk if you share with them. That would be lovely.
Oh yeah, maybe. I'll speak about it. I will if you want.
That would be great. Thank you.
And then I have a collaborator in the University of Verona.
All right.
She's also very nice, and she's also supporting me. And with her name is Dr. Antonella Menzi. We collaborate on this project because this is like a big thing that we sort of we're only part of it. So we're doing the neuropathology, but then some people will be doing other aspects of the research, you know?
Yeah. Such as?
Yeah. Such as something called, so for example, proteomics. So that's looking at proteins, and we'll be looking at proteins in the cerebrospinal fluid. So that's the fluid that surrounds your brain and spinal cord, and that sort of links to what we see, right? So if I see a very damaged brain, and we take, because these are all part of the brain bank in London, in Imperial College, and from the same case, that cerebrospinal fluid, or CSF, if we look at the proteins, are there like a specific signal compared to a group that doesn't have a brain as damaged, you know? So that's sort of analysis. there's like bioinformatics, biostatistical analysis. I do a little bit of that as well, but thankfully I have that collaboration as well where I can ask for help and learn about it. Yeah.
Wow. Do you also share some of your results with the people who you work with in the lab as well?
Yeah.
Is any other person who's working on MS as well with you?
With me, yeah. So the MS team is sort of the one in our floor in that lab.
Yeah.
And I have another PhD colleague, and she's very good at what she does as well. And then recently we've had a research assistant or a lab assistant, and she's also great, and we get along, and it's a good team.
All right. So what do you guys do after then? When you guys had a complicated research, things didn't go well, then do you guys in the lab join together to discuss this thoroughly and then trying new tests, to get the results?
Yeah. I would say though that it is not as like flexible in terms of, let's try this new thing. It's more like there is a protocol, right? So a lot of the labs, a lot of the groups, they follow protocols because they look at specific things that they have expertise on, right? So someone might be very good at measuring a lipid. Someone might be we are very good at staining tissue, right? So we follow specific protocols. If something goes wrong, then maybe we have to look at which step in the protocol it went wrong, or ask our supervisor, we've done this and that, There's a process of optimization as well, which that is, if we're trying a new antibody, because we stain with antibodies, we're trying a new antibody, we try different concentrations, we try different retrieval solutions, and then we sort of see then how it looks in the microscope, right? This one looks great, we've used this and that. This one doesn't, Yeah. And then once we know, the one that looks great, then we use the same steps for, I don't know, to stain 30 or 40 cases, So,
or maybe showing that result at a conference or an MS congress.
Yeah.
Well, did you go as well for conferences? Did you present them?
Oh yeah, I did go to one recently.
Right.
And that was a meta-analysis that I'm doing. And so that's more like what the literature says about the sort of things we're studying. And I was lucky to present, and it didn't go as well as I wanted, but I think it's just up from here. I think the next one will for sure be better.
Yeah. But tell me more. What didn't go well?
All right, I will say. So basically, my PowerPoint had like images that they're high quality because they're from like microscopes.
Or too much graphism. Too much graphism.
No, I don't know. I had to send it. I couldn't send it through email, so I had to send it through something they provided called WeTransfer, right? So I did that, and I think for some reason, maybe it was actually my fault because I was using some like template. The format didn't match. The format completely changed. So when I actually like stood there in front of everyone, and I clicked for the first time, I instantly saw that some things were off. So some images were like smaller, some text was different and like moved. So that freaked me out. And I didn't know how, like, I was like, okay, well, these slides are not supposed to like be like this. And then I tried to carry on. And I didn't look at the audience because I was like freaked out. I just looked at my screen.
Yeah. I mean, which is quite acceptable.
It was worse in my head.
Yeah.
Yeah, it was worse in my head because I had like done like a script, right? And I was trying to like go through the script in my head. And what something I'll share is that because usually, right, I'm very relaxed. I don't do scripts, but for this one, because I cared, I was like, oh, I'm gonna do a script. But actually, what I realized is that because I had this script in my mind, right?
Yeah.
When something didn't go as planned, I sort of freaked out. Whereas I recently did my second year review, right? And I made my presentation. I knew what I was talking about, but I didn't do a script because I know what I was talking about, right? And obviously the examiners in a review, they ask you questions to kind of know more about it, right? Or they might sort of, not tricky questions, but if you've done the job, then you should know this, right? Or you should be able to explain this, which is also something that, because I didn't have a script, I could just relax and sort of openly just discuss something. And basically, everyone is different. My style is no script, just know what you're saying and maybe have like a few. Obviously, you have the slides, you have to go through them, that's your structure. But more than that, I'm not gonna ever again learn something by memory, like a 10-minute talk by memory.
No, I do feel it, but I mean, congrats to you, because not many of us can't let go the script, because it feels our comfortable chamber, so to speak, where sometimes you're going to look in order to guide yourself of going with the flow, knowing what you're doing, even though you know what you do, you still you have that tendency to rely on yourself onto the script. But that's good on you. That's a great skill, and that makes the audience assume that you know the whole stuff, your whole topic, you know what you do. At the same time, after that, it's also a bit annoying having questions from the audience that they would expect that you know everything, where you actually learning from them and learning even more from from the analysis that you took on the pathology of the brain. It's a bit annoying, to be honest with you, with that. And sometimes you have to figure it out in the spot. What should I answer to this question that I don't know? Have you had that sort of thing? Lots of times, I'm sure.
I don't know. I mean, in that particular case, well, my supervisor told me, when you present yourself, say that you're a PhD student, right? Because if that conference, maybe, I don't know, doctors and like professors also give talks, I guess the questions for like a professor, they're a little bit more, or maybe harder, than for a PhD student, because we're just learning. We're students at the end of the day,
Yeah.
We're still developing that expertise. And I didn't have anything too bad. I think, sometimes maybe because everyone's a little bit biased. They know they have their own niche. Maybe they wanna, shift it to their niche a little bit, and they ask something that's really for them to answer, not for me, right? But in general, no. Like they're all sort of, they were nice. It was a good conference, and everyone was very, very.
Yeah, I think it also would depend on how the atmosphere is, and I couldn't agree more. Couldn't agree more with you, and to say that on my side, I also had lucky in having a good audience, where most of us were PhDs. A few of them were postdocs, but they were also learning, and that was quite rare to have lecturers hired by the university, where they were lecturing and throw some of these questions that seem complex. I mean, some of the questions that were thrown during one of my conferences from PhDs themselves, there was one that was really complicated, and I said what I said was, despite having not having extensive knowledge of that specific topic, because I'm training on this particular theme, what I could say to you very briefly was XYZ. But if you want something more thoroughly, I would suggest you to maybe check out this book, or maybe consult this lecturer who I referenced, and get a bit more get a bit more knowledge, and take a bit more from what the other one knows.
Yeah. I think what Dr. Menzi, my collaborator, told me is that I should just say, thank you for that question. That's a very good question. Unfortunately, I haven't thought about it, but I will consider it. You know.
Yeah.
And then that's it. That's like a safe pass.
Yeah, it would be a safe pass. So you mentioned me that you have 3 hobbies that you love: singing, playing guitar, and football. Which, by the way, did you see the latest signing of Colo Colo? You know, Vozinha is the goalkeeper. He was a great goalkeeper, and he used to play for the second league, in Portugal. And
Wow.
yeah, he last this season that has passed. He was still playing as a goalkeeper, but then before, when the season ended, he became a free agent. So I wonder, how was your reaction when you saw that Vozinha was signing a 6-month contract?
Colo Colo.
Yeah.
I wasn't expecting it, to be fair. I think, I don't know, because he's like 40.
Yeah.
I guess for keepers is different, you know. Like you can play even if you're a little bit older.
Yes.
But yeah, Colo Colo is I think is a good team. Even though Chilean football right now is not at its best, because you know they didn't qualify for a World Cup. And,
Yeah.
You know, so I guess I don't know.
Maybe
Maybe
miss.
he will increase the level.
Maybe, but I do miss the times of the Chile back in the early to mid 2010s, when Vidal, Alexis were in. They were 2-time in a row Copa America. I watched the matches. Do you remember that?
I checked with my colleagues. I think during that era, Chile were like number 3rd in FIFA rankings.
Yes.
Number 3. So that was big. But now, I think they're not that good.
Yeah. So they declined. Must be a reason from it.
A little bit.
I was curious as well here. Did you also heard, or maybe read somewhere in a journal about MS, and whether they identified a former football player who had multiple sclerosis? Do you have any knowledge of this, or have you heard of it?
No, man, I'm sorry.
I was just curious.
No idea.
Might be somewhere, but who knows? But yeah. But going back to the question, your hobbies: singing, playing guitar, and football. If you were having a stressful time, did you had this opportunity to sing in the lab, or maybe during your break, in order to improve your PhD research? So you're in a stressful moment and decide, I'm going to play a bit of guitar, sing a bit.
I don't know. I think actually there is a piano room in Fulton, and
Yes.
sometimes after work I would go. Or At any time I felt like, you know, just playing and singing. But now I sort of, it's not too bad, because if I'm stressed in the lab or in the office, because I do a lot of computer work, I can just speak with my colleagues, my friends, and they're it's a very good atmosphere. You know, before we said, atmosphere is very important in the office, in your workplace. Having a good atmosphere is invaluable, because you know you spend there like 8 hours a day or something. And
Yeah.
thankfully, I have a very good one. So if I'm a bit stressed or, I just speak to someone, have a laugh, grab some biscuits, or
Yeah.
go for a walk.
Yeah. No, that's
It's good
great.
environment.
Yeah, that also counts during the PhD. And if you're having a toxic relationship with your supervisor, or in a toxic relationship in the lab, then you are basically cooked.
Yeah.
Right?
Yeah.
But sometimes I think
can't-
yeah.
Yeah, no, just I can't be thankful enough to my supervisor. He's awesome. He's the best. He's amazing.
I mean, Swansea is a good reputation of having great supervisors and having a good great teaching staff across the majority of the departments, and they take the PhD obviously in a professional manner where they demand from you, but also they see they see their past versions, so they treat their candidates in a holistic manner and then prepare them for what comes after. Have you ever come across with these conversations with your supervisor of what you're gonna do afterwards?
Yeah, I think. Yeah, I'm grateful that my supervisor is a good one, and I do consider him also like a mentor. So we do have these conversations about, yeah, like how was it in your day, or what to do in the future.
Yeah.
What do we think about like this, it's really good, as you say, important to have a good relationship. Yeah, it's obviously essential for the PhD for you to enjoy the PhD.
Yeah. And then having the balanced life, playing football, singing, playing the guitar, doing other stuff. Doing other stuff. Do you also do any?
Very, very important.
Yeah. Do you also do other things outside of research? So, like, do you teach? Do you?
Not really, just
Mm.
yeah. I mean, teaching is part of my contract and my role, so
Yeah.
I do spend some time teaching, marking, doing some workshops, supporting in lab practicals, and then outside of that, just spend time with my partner and my cat.
Yeah.
And yeah, just drive around.
Mm.
And yeah, I mean, now holiday, like now it's August, so maybe we'll go to Italy for a little bit.
Okay, that's nice. Where are you guys going in Italy?
So my parents from Tuscany,
Yes.
and yeah, just Tuscany, which is lovely. So
Yeah.
complain.
It's a great region, and if I get an opportunity to travel there, especially to Firenze,
If
that
you
would be
love
nice.
art, yeah,
Yeah.
Florence is honestly so nice.
Yes. I had a teacher once who went to take his PhD in fine art and restoration of fine arts, and he told me much of his stories about Florence, from the Duomo till the stadium of the football stadium. And
Nice, man,
yes, and he said, if you get an opportunity, travel to Florence, and you will enjoy it much. So, yeah, I'm definitely will enjoy it.
yeah, for sure. I would say though, like it does get like, I don't know, 40 degrees or something crazy. So
Yeah, it is.
Yeah.
It is crazy, which I think.
probably go September, October. Is
Yeah.
better than now.
Yes.
I mean, I like my partner lives by the like close to the coast, so we go to the beach a lot.
All right. Which I think. I mean, I know 40 degrees is unbearable. I mean, from many of us that come from a southern background, who used to live in the south for quite a while, I think that the person can get used to it, even though
Yeah.
those temperatures also affects us.
Yeah.
So
Yeah, if you go, obviously you will enjoy. Just
yeah,
drink lots of water.
yes, definitely. Drink lots of water, definitely. Visit as much as museums and art galleries as possible, but always carry 2 bottles of water on each of your shoulder, on both of your shoulders.
Yeah.
So, would you like to share any other novelties about your PhD research? Any other challenges? Do you have any upcoming challenges as well throughout your research, besides the current one you are doing?
No, I just like to say that, in any PhD in like biomed or medicine or actually any field, just be curious.
Yeah.
Because you need that curiosity to like drive your research and find novelty and stand out and sort of create your own path as well.
Yeah.
And yeah, and also collaborate. I think obviously it's not up to everyone.
Yeah.
But I'm like lucky to be part of this big collaborative effort, and it's really good. Like you can feel that the quality is improved, not just the quantity, and it's good. So if you can collaborate with other people, I think you should, and it's great.
So that would be your advice for those who are listening and want to or are interested in undertaking a PhD in multiple sclerosis then?
Yeah, and also like know the literature, because obviously you can't know everything.
Yeah.
But reading papers is really important, and you do like learn a lot, and maybe like me and my supervisor, we say like the classics, like
Yes.
there's always a few papers in any field that are like really important. So learn, like just read those, and then slowly read things that you're interested about, and always be reading papers.
So if you have in memory, what was the first paper about MS you've read and you felt that this is what I want to do?
I think there's like a couple of very good reviews. I think one is in like the Lancet Medicine or something, but I can't. It's just called like multiple sclerosis or something, and then the author I can't really pronounce, but I could find the paper. And then there's a couple that we always go back, because I think one's called Pete et al. 2022 is something to do with the well, and then there's another one, Kuhlman 2020 or something. But there's two papers that are to do with describing MS according to the biology, not just. Do you remember when I said this traditional manifestation of symptoms?
Yes.
Of like relapsing, remitting. So that's like the traditional description of MS. There's different types according to like these kind of symptom patterns. These two papers are saying, actually, let's like describe MS according to the biology. And that's sort of what I do, right? What our lab does. So those two, very important, and they're like one of the first ones I read.
Yeah. So for the listeners then, collaboration and start reading papers are the stepping stones then for you to get your both feet into the door of multiple sclerosis then. So probably they will take that advice. And curiosity as well. Curiosity is very important, and that the PhD also motivates us to be more curious, asking questions, check everything. Obviously, you're not going to be perfect. You're the total expert, but you're always learning, and that's the beauty of it.
Yeah.
So, listen, man, it's been a great conversation. I would like to know, where can they follow you, where they can get in contact if they want to know more multiple sclerosis, or just simply reach out to you.
Yeah, I just say LinkedIn. I have Joic Majo. I think that's my LinkedIn. I'm open, yeah, to speak and discuss. And
Okay. So, man, thank you so much. We've finally made it. And I mean, if you want to do this again, I mean, let's go for it. Maybe after your PhD, and we could talk about any more of your results, how it went, and then the fever, and so on.
yeah, for sure.
Yeah. So, man,
Awesome. Thank you for having me.
no, my pleasure, Joic.
Great.
My pleasure. And for the students who are interested in studying MS, whether you want to go for a master's or in this case the PhD, just reach out to Joic and he'll be glad to have a talk with you guys. And even if you are in Swansea as well, for the Swansea listeners here, reach out to him. You can go to the lab. It's not too far. Maybe you're also researching a floor below.
Yeah.
So, Joic, once again, thank you so much. And for the students and graduates tuning into this session, thank you all for tuning in. Share it with a friend. And it's been a pleasure.